Welcome to this explainer. I am absolutely thrilled to have you here today because, well, we are unraveling one of the greatest scientific detective stories in human history. We're going back to April 1953, diving into the pages of the journal Nature to look at three short yet totally revolutionary papers that finally cracked the code of DNA. We're going to see exactly how these brilliant minds pieced together a puzzle that fundamentally changed our understanding of biology.
So how exactly did a bunch of seemingly blurry X-ray photographs and some pretty intense math unlock the literal secret of all living things? Let's dive right in. Today, we're looking at the clues scattered across these three groundbreaking 1953 papers. You've got one by James Watson and Francis Crick, one by MHTF Wilkins, A.R. Stokes, and H.R. Wilson, and one by Rosalind Franklin and R.G. Gosling. Together, they form a perfect map of discovery.
Section 1. The Mystery and the Flawed Model. By the early 1950s, the race to find DNA's 3D shape was totally on. Scientists knew its biological properties contained immense complexity, and that it was the stuff of genetics. But its actual molecular shape? Complete mystery. And in the mad rush to be first, guessing led to some huge mistakes. Take the legendary chemist Linus Pauling's model, proposed right before Watson and Crick. He suggested this three-chain model with the phosphates tightly packed on the inside of the molecule and the bases pointing outward. But Watson and Crick spotted a massive chemical flaw. Phosphates are negatively charged. If you pack a bunch of acidic, negatively charged atoms right next to each other along a central axis, they are gonna repel each other. The forces would literally tear the whole structure apart. Plus, the distances between the atoms were simply too small. Guessing just wasn't gonna cut it. They needed hard evidence. Which brings us to Section 2, reading the X-ray clues.
The solution wasn't going to come from building theoretical models in a vacuum. It had to come from actual experimental data, specifically shining X-rays through DNA fibers to see how the rays scattered, and then using those patterns to reverse engineer the molecular shape. Enter Rosalind Franklin and R.G. Gosling's crucial Structure B photograph, taken from calf thymus DNA. Just look at that striking cross-like pattern of dark smudges right in the center. To the untrained eye, sure, it just looks like a blurry X. But to the scientists analyzing this, it was a literal map. The complete absence of reflections on the meridian, the vertical axis, and that dominant cross shape immediately screamed one specific geometric form, a helix.
Now, of course, they didn't just eyeball it. The paper by Wilkins, Stokes, and Wilson backed this up with some incredibly heavy math involving things called Bessel functions. Don't worry, we are absolutely not doing calculus here today. Just know that Bessel functions are essentially the mathematical shadows cast by a helix. By mapping the squares of these Bessel functions onto the X-ray layer lines, the researchers mathematically proved that a continuous, uniform helix was literally the only shape that could produce the specific pattern of radiation. And looking at the conceptual diffraction pattern derived from these helices, specifically the empty regions, Franklin and Gosling noted another fatal flaw in Pauling's earlier model. See, DNA is highly hygroscopic, meaning it absorbs and absolutely loves water. Because of this, the heavy phosphate groups simply had to be on the outside of the helix, shielding the rest of the unit with a sheath of water. This is what allowed the central parts to safely take up their proper configuration.
With the phosphates confirmed on the outside, the X-ray photographs allowed the scientists to deduce the exact physical dimensions of the DNA molecule. Let's do a rapid-fire breakdown of the math, starting with this number, 34. The strong reflections on the layer lines of the X-ray proved that the helix has a pitch, basically the height of one full spiral turn, of exactly 34 angstroms. We are talking a measurement so unimaginably tiny it is on the scale of atoms.
Next up is the number 20. By measuring the angle of the helix and the position of the maximum intensities, they calculated the exact diameter of this molecular cylinder. DNA is incredibly narrow, measuring just 20 angstroms across. And finally, the number 10. The researchers saw a very strong reflection on the 10th layer line of the X-ray. This told them that within that 34 angstrom turn of the spiral, there are exactly 10 nucleotide bases neatly stacked upon each other, spaced 3.4 angstroms apart. They now had the exact architectural blueprints of the molecule. Section 3. Enter the double helix.
This is where the tension really hits its peak. Armed with these precise, undeniable mathematical measurements, a 34 angstrom pitch, a 20 angstrom diameter, 10 bases per turn, and the phosphates on the outside, James Watson and Francis Crick stepped up to propose a radically different and ultimately correct structural model. Their proposed diagram from 1953 shows not one, not three, but
but exactly two helical chains coiled around the same central axis. The ribbons you see symbolize the two phosphate sugar chains, and those horizontal rods, those represent the pairs of bases holding the entire structure together.
Watson and Crick laid out strict architectural rules for this double helix. First, both chains are right-handed helices. Second, owing to a geometric property called dyad symmetry, the sequences of the atoms in the two chains actually run in opposite directions, kind of like a two-way street. Third, as the X-rays proved, the phosphates are on the outside. And fourth, the nitrogen bases are on the inside, tightly packed at those 3.4 angstrom intervals. The Wilkins paper brilliantly compared these stacked bases to a pile of pennies sitting in the central region of the helical system, looking almost exactly like a spiral staircase. Section 4, the secret of life.
But this isn't just about beautiful geometry, right? The real genius of Watson and Crick's model was in how those horizontal rods, the bases, connect the two opposite chains to hold the ultimate secret of biological life. Because the phosphates are on the outside, the bases are forced together on the inside, and there's a very strict spatial rule here. The only way the hydrogen bonds fit perfectly across that tiny 20 angstrom diameter is if a larger purine always binds with a smaller pyrimidine. Specifically, adenine always bonds with thymine, and guanine always bonds with cytosine. So the absolute crucial takeaway is this. If you know the sequence of bases on one chain, you automatically know the exact sequence on the other chain.
Which leads us to what might be the greatest understatement in the history of science. At the very end of their paper, Watson and Crick dropped this famous sentence: "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." I mean, talk about the understatement of the century. Because the two strands are complementary, if you pull them apart, each strand serves as a perfect template to build a brand new identical double helix. They had literally just explained how life replicates itself. So we end right where we began, but with a new, even more profound mystery. We've seen the X-rays, we've walked through the math, and we've built the double helix. But I want to leave you pondering this final thought. How does this deceptively simple two-chain geometric structure, using an alphabet of literally just 4 letters, dictate the vast, mind-boggling complexity of all living things? It is the question that birthed the modern era of genetics, and one that scientists are still exploring today. Thank you so much for joining me on this explainer. I really hope this sparked your curiosity to keep exploring the incredible building blocks of life.